Hello, everyone. Today I will discuss the patent case for the Association of Molecular Pathology versus Myriad Incorporation. This case is related to genetics and DNA sequence, asking the questions, does gene belong to nature or companies? The Supreme Court case held in 2013 disputed the patentability of DNA sequence discovered by the Myriad Genetics Incorporation. BRCA1 and BRCA2 genes can predict a patient's risk for breast and ovarian cancer. This raises the questions, are gene sequence patent eligible? Also, to what extent are isolated DNA sequences a natural molecule or a product of human manipulation? To understand the concept of genetics in this case, let us briefly touch on some of the key concepts. In almost every cell of the human body that contains a nucleus, like 23 chromosomes. Chromosomes are a condensed, wound form of a long chain of molecules known as DNA. DNA is composed of phosphate sugar backbones and 4 distinct base sugars called adenine, guanine, thymine, cytosine, A, G, T, C for short. DNA serves the function of holding the instructions for RNA polymerase to create a transcribed mRNA strand that can be translated by the ribosomes to create polypeptide chains of amino acids that can be later formed into functioning proteins. Isolation of DNA is a tedious process in which scientists can work backwards to the previously explained process to figure out the sequence of the base pairs that compose genes, the functioning unit of DNA. Isolated DNA for BRCA1 and BRCA2 genes of this case is an 80,000 base pair long, including exons and introns. Exons are genetic materials that code for proteins, and introns are known coding DNA sequence. Scientists can synthesize an exon-only strand that can be used to mirror it and get a complementary DNA strand known as cDNA. Myriad Genetics Incorporation was founded in 1990 and found BRCA1 and BRCA2 genes in the years 1994 and 1995. They held 282 patents related to the BRCA1 and BRCA2 genes. This allowed the company to develop laboratory tests to detect patients high risk for breast and ovarian cancer. The Association of Molecular Pathology, was founded in 1995. The structure and leadership to emerging new field of molecular diagnosis. The Association of Molecular Pathology, or AMP, challenged Myriad Genetics Incorporation in 2009 for the isolated DNA of the BRCA1 and BRCA2 genes, claiming that is a natural product. To understand the challenge that they faced, let us look at the Patent Act of 1952, which aims to encourage innovation and discovery by allowing a legal monopoly over an individual's or company's new product or process. This Act of 1952 replaced the patent law in the United States Constitution dated back to the 1700s, initially installed by our founding fathers to protect inventors. In a section of the Patent Act of 1952, Section 101 specifically states that laws of nature, natural phenomena, and abstract ideas are excluded from patent eligibility. In 2009, Myriad Genetics Incorporation was challenged for 15 of the patent claims they had. This happened in the Southern District Court of New York by Judge Sweet. The decision then was the DNA is founded in nature, and Myriad cannot claim patent on the isolation of the DNA sequence and the cDNA sequence for BRCA1 and BRCA2 genes. Therefore, Myriad Genetics Inc. Went to the Courts of Appeal in 2010. They claim that not all the molecules in the DNA are founded in nature. They had to manipulate it somehow. The decision was that DNA is a molecule, and Myriad can claim patent on the isolation of DNA and cDNA of the BRCA1 and BRCA2 genes. The case was taken to the Supreme Court, the U.S. Supreme Court, in 2013. In this court, the Association for Molecular Pathology claimed that the isolation of DNA had no human manipulation and therefore does not warrant a patent. Myriad claims, on the other hand, are that not all the molecules in the isolated DNA and cDNA are found in nature. They had to be manipulated by humans in order to obtain this information. The decision of this court was very important. DNA is a naturally occurring molecule, and although difficult to isolate, it does not warrant a patent. cDNA, on the other hand, involves human manipulation of the extraction of introns and exons, and thus deserving of a patent. The outcome of this case is very important. 10 years after this year, is 10 years after the Association for Molecular Pathology versus Myriad Genetics Inc. on the DNA patentability case, is the ability to treat, gave the ability to treat and diagnose diseases. Was revolutionized at the time because it allowed other companies to use the information for the isolated DNA sequence as it was no longer patented. The court's decision opened the door to innovation, allowing science and research to keep moving forward. Here are my reference list for all the information that I've gathered. I hope you liked my presentation. Thank you very much, and you have a wonderful day.